Hi everyone, welcome to week number 2 in our Bio 120 lab. Can you believe it? We're already finished with our first week and we are into the term now. So let's see what we've got going on. So last week you learned about the scientific method, how science is actually done. It's a little more complicated than what you learned about when you were in grade school. But it is a tremendously good and effective method for looking at the natural world. One of the most important parts of it is peer review. Peer review is tremendously important because it keeps everyone honest and it verifies results that other scientists have gotten. So it's a terribly important method. You did some graphing last week. You looked at examples of wheat plant heights and rye plant heights, and you graphed those data on a graph. You did linear regression to look at the lines. You used the equation to predict what would happen at other times during the experiment. You also graphed your data from the Alka-Seltzer experiment, where you looked at different temperatures of water and measured how long it took your Alka-Seltzer tablets to dissolve, and you graph those results. So this is good practice for you because a couple of times in the term you're going to be using graphing, so don't think you can just forget it because you're going to need that skill. And remember, you can always go back to those instructions in lab number 1 to help you with Excel. All right, so what are we covering this week? We're covering some chemistry. And some of you will think, oh no, why are we studying chemistry? This is a biology class. Because all of life depends on chemistry. So organisms are made of molecules. Molecules are made of atoms. And so you have to know something about, especially molecules, when you're studying some biology. Like, why is a protein so important in organisms? And what happens when you denature or unfold all the proteins in an organism? Well, I'll give you a hint, the organism's going to die. So it's really important to understand the basics of chemistry. We're going to concentrate this week on pH and buffers, so we need to talk about those for a minute. What is pH? pH is a measure of how acidic or alkaline an aqueous solution is. And aqueous means water. Well, what determines when an aqueous solution is acidic? When you add more hydrogen ions to a water solution, it becomes acidic. Hydrogen ions are basically just protons. If you add hydroxide ions, you get a more alkaline aqueous solution. Well, why is that important? Because almost all organisms, almost all of them, love a nice neutral pH where there's a balance of hydrogen ions and hydroxide ions. Notice what they look like. When you add this plus this, what do you get? Water. Exactly. And so hydrogen ions and hydroxide ions are what form when water molecules break apart. And you want equal numbers of those. Why is an acidic solution not good for organisms? Because you have too many positively charged hydrogen ions and they react with all the different molecules. Why don't organisms like alkaline solutions? Because there are too many negatively charged, negatively charged hydroxide ions that react with your molecules. So most organisms like a nice neutral pH of 7, and that's where there's a balance of hydrogen and hydroxide ions. You'll also be taking a look at buffers. What are buffers? So buffers are chemicals that help to maintain a certain pH, and they don't have to maintain a pH of 7. They can maintain a pH of 4. It depends on what the chemicals are. But anyway, buffers are used to maintain pH. For example, in our bodies, we have buffering systems in our blood so that our blood doesn't become too acidic or too alkaline. Remember, we're organisms, so we like a nice neutral pH. So you're going to take a look at a buffer, sodium phosphate I think we have in the kit, and how it actually buffers when you add acid or base to it. As a word of caution, be very careful with this lab because you are working with acids and bases. So be careful around children, be careful around pets, be careful of spilling these solutions, wear your safety equipment. So that's safety cautions for you for this lab. Review the introduction and the overviews for Module 2 to help you understand a little bit of chemistry. They will help you. Your lab instructions, remember, are found in that introduction. Gather up your supplies. Labeling is very important. You're going to label the little wells that you have so that you know what everything is when you're performing the experiment, and so that I know what they are when you take pictures of your results at the end. Perform the experiment and record the results. You'll be taking a couple of pictures this week, and you will include those in your lab report. They are not separate. You include them in the specific places in the lab report. One thing that I don't think the instructions tell you very explicitly is to mix your liquids after you combine them in a little well. A little well is just a little compartment in the little tray. So make sure that you combine your liquids once you put them together in a well. You can pipette. Pipette is just like a little squeezer dropper. Gently pipette the liquid up and down several times before moving on, and this will help to mix your solutions. Then you're going to write your lab report, include your photos, and this is due by Sunday evening at midnight. All right. This week you will also be starting your final project. So your final project is worth more than 200, can't remember exactly, more than 200 points out of 1,000 points for the term. So this is a big project. This week you are going to be choosing one of the experiments to do. Look at this section under Module 2, and when you click on Final Project Guidelines and Rubric in this section, it will have a link there for the Ask a Biologist website. And if you go to that website, there are 4 experiments that have already been chosen for you by SNHU that you can do. Oh goodness, let me remember. Caterpillar, nervous experiment with the paper clips, the air pollution, and the water. Yeah, those are the 4 that are listed for you on the guidelines and rubric. And they can be found at Ask a Biologist website. If you choose to do one of these experiments and modify it, then you don't have to get approval by me. If you want to use a different experiment, if you have a different idea for an experiment that you can do for this term, and then you're going to write it all up, give me the significance of it, present the data to me, all of that, tell me what it all means, then you have to get it approved by me. There are a couple of other ones. Hang on, where did I write them down? There are a couple of other ones on the Ask a Biologist website. Breaking proteins, that's a really good one. All you need are egg whites. Really cool. Really cool one. If you're into cooking at all, this is a really good one to do. So breaking proteins is a really good one. And heavy water, that's also another really good one that's from this website. Now, you can think up something on your own if you want to, but one thing that you have to remember is that you have to be able to get this whole thing finished and a presentation made by week 7. So, growing plants over the summer, that's not going to work for us because you can't get it finished in the time that we have. So remember that your research has to be doable, and we have to get it finished and the presentation finished by the 7th week. So I just want to make you aware of that. If you have any questions about this, if you want to ask me, is this a good one, that a good one, please, please, please email me. I'm here for you. I can help you with lots of different things, your understanding of the material, any questions that you have about the experiments, because I've done them, I will be doing them all along. So just ask away. I am here for you. So let me know if I can help you with anything, and I hope you have a wonderful week.